introduce Dr. Antoine Snijders, who's here to present at the Interdisciplinary Instrumentation Colloquium. This is the start of a new set of organizers, and I'm one of your organizers, Jamie Inman. And so it's really my pleasure to introduce Dr. Snijders, who's here from bioengineering and biomedical science. He is the department head and here at LBNL, and his research goals are to understand the complex interactions among genetic background, environmental exposures, and the microbiome in determining disease risk. And he's going to tell us about the Bella petawatt laser platform, laser proton beamline, a new platform for ultra high dose rate radiobiological research.
Thank you, Jamie, and welcome everybody. It's a real pleasure to be here. Okay, so as Jamie said, I'm going to be talking about some experiments we've been conducting at the Bella petawatt beamline, exploring the radiobiological effects of ultra-high instantaneous proton dose rate regimes. But I first wanted to start off with a little bit of background of the lab. There's been a long history in the development of radiation therapy here at the lab. Of course, Dr. Lawrence came to Berkeley in 1928, where he invented the cyclotron in 1929. He opened what was called the Red Lab at the time in 1931. And together with his physician brother, John Lawrence, and a host of other scientists here at the lab, built increasingly larger cyclotrons and expanded the use of those machines for not just unlocking the secrets of atoms, but also for creating radioisotopes for medical research. And of course, in 1939, Dr. Lawrence won the Nobel Prize in Physics for his invention, and together with his brother, John Lawrence, established the Donner Laboratory, Donner Biomedical Lab at Berkeley Lab, and that really was the birth of the biosciences area as we know it today here at the lab. You see a picture here of the construction of the 184-inch cyclotron, which now houses the ALS. And the first beam was created there in November 1st of 1947. I do want to spend one slide on Dr. Wilson as well, who was here at the lab. He enrolled as a graduate student in UC Berkeley in the early 30s, and then joined Ernest Lawrence's radiation laboratory afterwards. And he's the first to propose the use of the Bragg peak for radiation therapy. That was in 1946. So the Bragg peak is making use of the stopping power of particles, where they release most of their energy at the end of their range. And if you focus that peak inside of the tumor, you can deliver really high doses of radiation to a tumor, and while at the same time reducing or delivering minimal dose in front or behind the tumor to normal tissue. That was in 1946. Then Professor Tobias, right after, said, okay, well, that's very interesting. Let's do some biological experiments here at the lab using protons in 1948. Then in 1954, the first humans were exposed here at the lab, patients with cancer. And approximately 1,500 patients were treated here at the lab during the time from 56 to 86. A side note is that Professor Tobias also predicted light flashes by cosmic rays long before they were actually observed by astronauts. So if you're looking straight into the path of a beam, you can actually see these, some people, these little light flashes or blue sparks or lines coming through your retina. The mechanism of which is not fully understood, but it was a thing that was done back then. You see Professor Tobias on the left, and on the right is, what's his name? Macmillan is looking in the beam right there, and it'll come to me in a second. Buttinger, Dr. Buttinger is on the right. So
it's not recommended to look in the beam.
it was not recommended, and I'm not sure if EHS was around at that time, but it's not something that should be done. But it was, of course, you had to show, you know, look here and you'll see light flashes. Yeah, not recommended. Fast forward to today, we know that radiotherapy is the current standard of care for more than 50% of all cancer patients. As I showed you the Bragg peak effect, so the charged particle beams, they do have a much more favorable depth dose interaction And that's shown here on the diagram on the right, where for X-rays in green, you have a very high entrance dose into the body, and then it's sort of you're losing energy as the X-rays move through the body. Using the Bragg peak and later on the spread out Bragg peak was developed, where they were able to spread that out a little bit so they could get the whole tumor irradiated with that high intensity beam, and still delivering little to very little dose in front or behind the tumors. However, we do know that access to that type of therapy, you can say, well, why aren't we all using proton beams for treating our cancer patients? Access to those facilities is limited, and that's mostly due to size and cost. These are enormously big gantries and accelerators and have a very high operational cost as well. So the motivation for some of our studies in collaboration with ATAP were that more compact accelerators were needed to increase access. However, even with the use of ions, normal tissue toxicity always limits the therapeutic window at which you can irradiate a person. So clinicians always try to deliver as high a dose as possible to a tumor without causing irreversible tissue damage, essentially. And that window is called the therapeutic window. And what we're presenting you today is work with our colleagues at ATAP, where we not only trying to increase that therapeutic window, we also trying to make more compact, cost-effective accelerators. I do want to introduce in one slide real quick the flash effect, which is well known in the radiation oncology field. It's really picked up in the last few years. And this is where you irradiate a tumor at dose rates that are much higher than those currently used in the clinical context. And when you do that, you reduce the effect of radiation on normal surrounding tissue. So you have much less damage to surrounding tissue. You have an equal response to the tumor. It doesn't matter to the tumor what dose rate you deliver this beam at, but the normal tissue effect is much less. And that's shown here in a schematic or in a cartoon for a mouse with a tumor. Conventional dose rate, you have quite a lot of surrounding tissue damage when you deliver that exact same dose in a flash regime, and that's defined currently as 40 gray per second or higher. Whereas a conventional dose rate is more like a gray a minute or so. So a radiotherapy fraction of 5 gray, 10 gray would take 5 to 10 minutes. At 40 gray per second, you could get all that in a second or less, that dose for that regime. That flash, it's termed the flash effect, has been demonstrated in bacteria, animal tissues, human cells, and a limited series of human clinical trials. And in nearly all of those cases, when delivered, radiation doses are delivered at flash dose rates, you have less damage to surrounding tissue. That was the flash effect was actually first identified. It wasn't called the flash effect back then in 1969, where people started to realize that if I irradiate cells, these were human cancer cells, at 2 different dose rates. So they use a cobalt-60 source to get 100 rad per minute. So it's a gray per minute, standard conventional dose rate. Or they used their beam called Plato, which was able to deliver a 7 nanosecond pulse. So deliver that same dose in a fraction of a second. And what you see in the data is that the surviving fraction of these cells is on the y-axis for increasing doses on the x-axis. So as you deliver more dose to the cells, fewer cells survive. But if you deliver the dose in the black dots at a much higher dose rates, you get an increasing number of cells surviving. And that sort of was very dormant in the field. That effect was noted but not acted on until about 2014, when the laboratory of Dr. Catherine Vosenin in Lausanne, Switzerland, published a study in Science Translational Medicine where they irradiated the thoracic cavity, so the lungs of mice, with 2 different dose rates. They used in red a conventional dose rate, 1.8 gray per minute. In blue, it's 3,600 gray per minute. And then they looked at, on the y-axis, the percent of mice that had pulmonary fibrosis. So that basically is the formation of scar tissue in the lungs. That is a common side effect of radiation of people of the thoracic cavity. It makes it harder to breathe. It's a common side effect, and clinicians try to avoid it at all costs. So what you see, of course, is this huge difference that at 30 to 40 weeks after exposure to the same dose, they were irradiated with electrons in this case. You see that the percentage of mice that came down with pulmonary fibrosis or that were diagnosed with that histologically afterwards was much, much lower. And of course, our colleagues at ATAP were like 3,600 gray per minute is great. Hold my beer, we can do 10 to the 7 gray per minute here. So let's do some experiments and see if we can push that down even further using laser-driven protons. Of course, the laser-driven protons have, the way we use them at ATAP are spread out based on, and I think I have it on the next slide, based on the repetition rate of the laser, and which currently is at a maximum of 1 hertz. So once per second, you get a shot of protons with a very a small time window. So you get very high dose rates up to the 10 to the 7 gray per minute. And then there's a little bit of nothing, and then there's the next shot of the laser pulse, and then so you get this pulsed proton beam that's being delivered. It's quite different from a conventional proton beam, which is at the megahertz scale. So the mechanism of that flash effect are not well understood, so that's why we were very interested in doing some of these studies using laser-driven protons. And this slide actually shows you how at the Bella Center we generated a proton beam from a laser source. So you see the laser interacting with the target. In our case, this is a very thin piece of Kapton film that then accelerates hydrogen and protons at the back end. And so the protons start streaming out the back. They're captured. And these protons have a very broad energy range. And as I said in the previous slide, the proton bunches are spaced out depending on first the repetition rate of the laser, which is about 1 hertz, and also the replenishment rate of that target. We had that on a spool, so we were spooling through the Kapton film with every shot of the laser. But we were getting very high dose rates with that, and I'll show you a diagram of the exact setup in the next slide. So I'll first talk about our work on Bella IP1 or interaction point 1, which is a large focal spot where we used an active plasma lens for proton transport. We irradiated cells, human cells with this, with TNSA protons at about 2 MeV. And then I'll show some work that we did using the IP2 or interaction point 2, which is a smaller focal spot and where we use permanent magnets to transport the proton beam. And for that, we actually did some in vivo mouse ear skin irradiations at a slightly higher energy at 7 MeV. So the standard setup at the Bella petawatt is dedicated to laser electron acceleration, where a laser beam interacts with a with plasma essentially, and which is then guided by a discharge waveguide to generate an electron beam. This setup is quite modular, and we were able to fairly rapidly, using an LDRD, turn this into a proton beam line, which is shown here from the top. You have the interaction. When we're showing the interaction of the laser with the Kapton tape is right here. You see a picture of the Kapton tape with the holes in it from each laser shot at the bottom. And the proton beam is then propagated down to our cell holders, which are sitting right out of the vacuum of that beam setup. You see the beam profile. These were taken about 5 centimeters behind the tape drive at different energies. So this shows how we go from proton acceleration to delivering protons on human cells in less than 2 meters. So first we have the interaction with the tape drive. Protons are then transported using an APL or an active plasma lens, which is in this case 33 millimeters in length with a 1 millimeter core in the middle, filled with argon. We applied a discharge on that APL of 90 amps to focus those protons, use a dipole magnet to clean it up from neutrons and electrons, an ICT to look for beam stability. It was an online diagnostic that was put in place, and then protons are delivered out of the vacuum on our cells. And behind our cells, we basically get gaffchromic film and a scintillator screen for the symmetry after the fact, essentially. We were then able to accumulate shots. So if we wanted to deliver, say, 10 gray, we would fire 10 shots. We had about a gray per shot that we knew of at that time. If you wanted more dose, we just accumulated shots on target on our cells to get to delivering different doses. The pulse length is about 30 nanoseconds of the protons with an instantaneous dose rate of about 4 times 10 to the 7 gray per second. As I said, we fielded an ICT in this experiment just to make beam stability measurements, but we also used that. We calibrated the charge that was detected on the ICT with the radiochromic film dose estimation at the back end. And so we were able to draw a curve on that, and we found that to be quite linear, which was very exciting because now we had an online ICT measurement, which is an instantaneous readout after each shot, or near instantaneous, I suppose. That would give us basically an estimate of the dose that we delivered on target without having to wait for the RCF to develop, which takes 24 hours, and then doing the measurements after the fact. So that was a paper published by Laura Gerlich in 2022. I think I got that. So one of the issues was that the energy of our proton beam was sort of in the order of 2 to 7-ish MeV at the high end. So we had to, just using standard tissue culture dishes, which are plastic dishes that we grow our cells in, that plastic would be too thick for the protons to actually go through and irradiate our cells. So we designed these cell culture holders here at the lab in collaboration with ATAP and their machinists to culture our cells on a mylar membrane. So it was 3.6 micron mylar membranes that we stretched in between. Basically, this middle part is a stainless steel plate, and then we had 2 small aluminum cover plates. And we basically stretched mylar on both ends, so we had a window to shoot through front to back, and so we could irradiate our cells on the front part of the mylar. The beam would go through, and we would then do the measurement using RCF and scintillator at the back end. And that worked pretty well. So cells, when you culture human cells, they need to be in liquid, in a growth media. So we wanted to get rid of that media during irradiation. So we designed this little cavity in the middle that right before we irradiate, we would flip these holders up so the liquid would drain down. We would shoot through, irradiate the cells, and then would flip them back down. And that's shown in the next slide, where we were able to use a basically linear stage where we would mount 7 of these holders, all hanging down at say a 45-degree angle, so the liquid would be on the cells. Cells would be happy. Then right before radiation, they would ride up this little ramp, and they would liquid would drain down. They would get irradiated, and then it would ride down the ramp on the other end. And that's basically shown on this side, where we had all these cells on the holder. So then after radiation, we collected our cells back. This was a 56 cell sample, 2-day, 1,000 petawatt shot experiment. So it was a big experiment for us. The cells we used in these holders are PC3 and RWPE1. RWPE1 is a normal prostate cell. PC3 is a prostate cancer cell. So we were able to compare what is the survival rate of the tumor cells versus the normal cells when we irradiate using laser-driven protons. And the survival we determined using a technique called clonogenic survival, which is a standard technique used to determine radiation sensitivity of cells. So after radiation, we would collect the cells, we would plate out in triplicate or more usually known quantities. So we'd count the cells, and we would put down 100 cells in a plate. We would then wait 3 weeks, stain those dishes, and we would count how many cells survived. So if you put 100 cells down, if you don't irradiate, you should get 100 colonies, essentially. So you would have 100% survival. And then if you irradiate with increasing doses, you get, as you see in these curves, you get less and less survival. So that's how typical survival curves are drawn for radiation sensitivity. And so we did that for the PC3, the tumor cells, the normal cells. after radiation with laser-driven protons at Bella. And then we had a reference X-ray radiation, the standard dose rate X-rays. And the X-rays are shown on the right. The tumor cells are in orange and the normal cells are in blue. The ones on the right are sort of your typical survival curves where they have very similar radiation sensitivities between those two cells. The normal cells are a little bit more resistant than the tumor cells, and that's well known for those cells. On the other hand, with the laser-driven protons, we see that there is quite a bit more survival. The tumor cells seem to be dying at about the same rate as compared to the reference X-rays, but the normal cells seem to have much increased survival even at higher doses, suggesting that, well, maybe we are seeing what's called the flash effect in vitro. We were then interested in, well, what is the biological mechanism of this? And so we did a sequencing study where we sequenced the transcriptome, which is basically the RNA transcripts that are in the cells. And so RNA is sort of the intermediary between the code in the DNA and proteins once they're fully formed. So the RNA levels gives you a readout of what the transcriptional state is of these cells at the time of radiation. Maybe that gives us an indicator of what the mechanism is. Why are these cells responding differently? You can quantitatively measure these RNA levels, these transcript levels. And when we did that, we saw that there were quite a few genes, and I'm just listing a handful of these here, that are at increased expression. So there's more transcript levels in the tumor cells, and there's fewer of those transcripts in the normal cells. And a lot of these genes are associated with apoptosis and response to oxygen. So oxidative stress seems to be much higher in the tumor cells and apoptosis as well compared to the normal cells. So that may point to some mechanism where, well, perhaps oxygen levels are really important in this observed phenomenon. And I'll show you some data that we did using a much more rudimentary approach. We used peptide irradiations at the ALS, where we also looked at the oxygen effect. There were a number of limitations, of course, with that study, and I do want to mention them here real quick. You might have seen that the error bars were quite large in our laser-driven work. That was mostly due to shot-to-shot fluctuations and sample-to-sample fluctuations in the actual dose that was delivered. We didn't have a good readout at that time, where we used the instantaneous data from the ICT to estimate the dose that was delivered. We have that worked out now and get much tighter error bars on the dose that is prescribed and the dose that's actually delivered. We use X-rays as a reference because we don't really have here at the lab a clinical proton dose rate reference. So we didn't have that at this time, but I think X-rays and protons are relatively comparable. The RBE, or the relative biological effectiveness of protons compared to X-rays, is about 1.1. So they are very similar. And of course, we wanted to do more cell types, and we wanted to do some in vivo studies. And that's what we focused on with our next experiment at Bella, where we used a new beamline, the IP2 beamline, which had permanent magnets, as shown here. Similar strategy, we still use the target sheet normal acceleration to accelerate our protons. No APL this time. We had a permanent magnet quadrupoles to focus our protons. Still had a dipole in place and several ICTs, which I think are on the next slide. 3 ICTs in this case. And then we had anesthetized mice. So these mice were alive, and we were irradiating a very small part of the ear, about 7 millimeters of the ear, with a very homogeneous dose that was delivered to these mice. This is a picture. You'll see a picture of the mouse here. This mouse is anesthetized and has an ear clamp. Essentially, the ear is clamped with a little hole in it. And you can see that on the picture on the right, so that we're radiating 7 millimeters of that ear very homogeneously, and the rest of the mouse is shielded with upstream shields. So you can sort of see the vasculature in that ear. We backlit that, so you can see that very nice. It's very thin. The mouse ear is about 250 microns thick. As I said, we had several ICTs worked in this setup for, again, for online dosimetry, but also for beamline diagnostics. We used ICT3 in this case, which is the one sitting right here. to help us with the online dosimetry. So we get a readout with each shot of the dose that was delivered. So this time we had it calibrated before the experiment. We knew exactly what the dose was that was delivered. And you can see that we prescribed 50 gray of protons, and we were pretty spot on for most of these animals. This was a 4-shot day experiment with 1,220 shots and 60 animals that were irradiated with different doses ranging from 10 to 50 grays of protons. And we use ear thickness and reddening as our biological endpoints for this study. And so this is the preliminary data that we got from this experiment. On the top, you see 3 panels. Those are all irradiated at Bella. On the left, you have the sham, so they received no irradiation. As I said, the thickness of an ear is about 250 microns. And it's pretty even across the time frame. There's not much fluctuation in that. I mean, we delivered 36 grays of protons. You see a little hump. It should occur about 20 days after radiation is when you get the maximum thickness of the ear. And when we deliver 50 gray, we see a much stronger response. And we see some mice that had a really strong response. We don't quite understand yet why there is such a variation, or why these 2 mice had ear thicknesses that were approaching a millimeter for some time. Most of these ears then after the 20 days are recovering. And so the mice recover quite nicely. We did some X-ray reference radiations, which you see at the bottom. For this, we used 2 doses. We used 36 gray and 40 gray. The 36 gray matches exactly the middle panel of the Bella protons. We also did a 40 gray radiation, taking into account the difference in RBE between protons and X-ray. So if you RBE correct the 36 gray times 1.1, you get about to the 40 grays of X-ray. And if you just do the comparison, the RBE corrected comparison, you can see that when we deliver the same dose with X-rays, we get an increased response. We quantified that. You see that on the left here. We basically using a t-test comparing the X-ray irradiated at 36 gray and 40 gray in orange to the Bella protons at 36 gray in green. So comparing to itself, the p-value is 1. And then with increased significance, the p-value of 0.05 is the dotted line here. Some borderline significance with the 36 gray, but very significant increase in thickness with X-rays at the 40 gray compared to the Bella protons. The sparing factor we calculated, which is the how much less damage is there. So when we irradiate with X-rays at 40 gray, we have a sparing factor of about 1.4. There's about 1.4-fold more damage with X-rays compared to laser-driven protons, which is great, great news. So we're trying to repeat these experiments in our current upcoming campaign. And we're also interested in studying the biology behind these observations. And that's something that we've started doing using, for example, infrared imaging here at the lab. It's a technique to identify the chemical changes that occur in biological samples. It's very rapid, it's label-free, it's non-destructive. And this has been done by Ho-Ying Holman here at the lab using her ALS beamline. She had looked in the past at some low-dose irradiated cells in this case, and found that there were changes in their FTIR profiles, their infrared profiles. So we did this, developed this this technique, the FTIR technique, on live animals here at the lab. This was done using T-Rex funding from IARPA, and where we were able to anesthetize a mouse and put the ear of that mouse under an FTIR microscope and basically measure with this germanium tip, measure the FTIR profile across a small region of the ear, non-invasively, non-destructively, and fairly rapidly. And that is work by Jamie Inman, and that paper is hopefully accepted soon. We then analyzed those spectra. So now what you look at here are the Bella laser-driven protons. We measured the what's called direct as the ear that was actually irradiated. The indirect is the ear that was not irradiated, so the contralateral ear. And then there's controls, which are the unirradiated ears. And he's showing you a PCA of the spectra obtained from these different groups of mice. And one of the things you can see is that we had a training set, we had a test set, and we found that there to be a significant difference, not just between irradiated and unirradiated, but also between the the unirradiated ear in an irradiated mouse and a control mouse. So that would be the indirect effect. So they're suggesting that there might be a systemic effect, that even though you irradiate this ear, you might still measure a signal in the other ear, even though that was not irradiated. And that's something we're quite interested in, what that is and what the actual chemical changes are. We haven't quite gotten there yet at this point, but it's quite an intriguing finding. Okay, so I wanted to spend a little bit of work on, or a little bit of time on the work we've done with Dr. Corey Ralston here at the lab, at the ALS, where we've been doing these studies in vivo, we've been doing them in vitro, but we really want to know how does flash actually work. And it's good to know that something works, but sometimes it's better to actually know how it works. And the reason for that is, first of all, there's several different hypotheses that people have come up with to explain the flash effect. One of them is patient motion. Patients breathe in and out during radiotherapy, and people saying, well, maybe that is what is causing some of this normal tissue damage. And with flash, you would deliver it in a second, so you wouldn't have that effect. Well, that may be true, may play some role, but it may not be the whole story. Differences in immune response. There are some very interesting papers now that show that when you radiate at a flash dose rate, you get a different recruitment of immune cells compared to conventional dose rate radiation. Tissue oxygen levels appear to play an important role, but it's still up for debate, and there might also be differences in the response to damaged DNA or damaged protein if you deliver it with flash or conventional. So there's different hypotheses, but it's important to understand the actual mechanism of the flash effect, because we know not all cancers are the same, not all people are the same. So if we understand how it works, we can maybe tailor that to different tumor types, different people, to get the maximum advantage of the flash effect for different people. And so what we did with Corey is we used a very reductionist approach. We said, okay, we're just going to use very short peptides, just a string of amino acids. They're going to put them in water, and we're going to radiate them at the ALS in this case. And this is a technique she often uses from the X-ray footprinting field, where people look at oxidative damage and then infer 3D structure of peptides. Because we know that when we irradiate water, we generate lots of hydroxyl radicals, and that's the OH, and the modification by OH on an amino acid is covalent. So if you look at a phenylalanine that gets attacked by a hydroxy radical, you actually end up with an additional 16 Dalton in size because of that oxygen that now got covalently bound to that amino acid. So you can then imagine if you radiate peptides at different doses, we measure how much of that total amount of peptide is actually modified, just have that 16 Dalton addition, and you can measure that by LC-MS. You just run it out on a capillary and measure the amount of modified versus unmodified peptide. So the increased size would be here, and then with increased doses, you can calculate. You see some real traces of unmodified peptide on the right. The black peak, the red peak is the modified peptide with the addition of the oxygen, and you can calculate the percent modification over dose essentially. So we did this experiment where we used different types of peptides and irradiated them at the ALS on beamline 331, which is a focused X-ray beam in the 1 to 16 keV range, and has dose rates that are way higher than 40 gray per second. And so we were able to expose them to microseconds to millisecond type exposures there. And I'll go into this setup a little bit in the next couple of slides on how we did that. This is Corey's jet flow, where basically we had the peptides loaded in the syringe on the top. We were able to push them through this capillary, and a liquid jet X-ray beam comes in here from right field and irradiates these peptides midstream. And then we determined the exposure time by the sample speed. So we were at different speeds at which we could empty this capillary. This shows you a table of how we got to the different doses that were delivered. So we delivered doses down to about 1.6 gray, up to 21 gray. The dose rates are extremely high. They are in the tens of thousands of grays per second. And the exposure times were either 90 microseconds or 450 microseconds. And we had some aluminum attenuators in place to allow us to get to different doses. So we did irradiations at the ALS. We did our irradiations with standard conventional dose rate X-rays at our X-ray 320, so the 1.3 gray per minute, and compared the amount of damage for different doses. And one of the first things for this relatively short peptide, the sequence is right here. If you look at the amount of the percent modified, so the percent of peptide that has this additional 16 Dalton on it, you see that when we irradiate with same doses at the ALS at these extremely high dose rates, you have much less damage compared to the X-rad, the conventional dose rate radiations. We also asked whether oxygen plays an important role in this. So these were radiated at the ALS, either in ambient oxygen, so saturated oxygen levels, or in very low oxygen levels, where we bubbled nitrogen through these tubes with the peptide and sort of got the oxygen level down to very low, ran them from the foundry to the ALS before the oxygen could get back in, and then irradiated them and analyzed them right away. So oxygen is obviously an important modifier of that effect. We also worked with our collaborator, Dr. Vosenin in Switzerland, and she was able to irradiate using electrons. This was a pulsed electron beam, and was able to deliver flash dose rates and all the way down to conventional dose rates, so 0.2 gray per second, all the way up to 10 to the 6 gray per second. And she was sort of able to see this stepwise decrease in the amount of damage just by increasing the dose rate at which the same doses were delivered. These were all delivered at 10 gray or 15 gray of electrons in this case. That ends my talk. So I have a brief summary here and then an acknowledgement slide. So in summary, we've developed several capabilities at ATAP to irradiate not just cells, but also animals on IP1 and IP2. We were able to show that for the in vitro work, we see increased survival for normal prostate cells compared to tumor prostate cells. So we're recapitulating the flash effect in vitro. We're seeing some very promising results in vivo on the ear thickness work, where we are seeing that we have less damage to normal tissue, normal skin tissue in the ear. We feel that the ICT on both experiments, and this allowed us to get a pretty precise application of the prescribed dose, 40 to 50 gray, on those ears. And now we are, of course, interested in going into higher doses of protons, going to 30 MeV protons, so we can do some in vivo tumor work, where we would implant tumors right under the skin of a mouse, like a xenograft model, which is often done in mice, and then we can irradiate that with 30 MeV protons, and then look at the normal tissue effect and the tumor sparing effect, or the normal sparing effect and the tumor effect at the same time. So that's sort of the parameters of the laser system that we're trying to shoot for going forward in the next several years. Of course, we want to go to 50 to 100 MeV, which would be much more clinically relevant. Clinical relevance is sort of 100 MeV and higher for protons. So this is a picture of the IP1 beamline. You see Jing-Hui Bin, Laura Gerlich, and Kei Nakamura at the ATAP Center carefully looking over our cell samples when they were being irradiated. And with that, I want to thank, there's a lot of people on here that I need to thank. I'll focus on the picture first. That is from our last campaign on the biology team. We got Dr. Mao on the left with Dr. Inman next to him. On the front row, we got Jared Deschamps, Laura Golik, and Sahel Hakimi. And in the back from ATAP, we have Kei Nakamura and Lieselotte Obst-Hiebel, who've been instrumental to getting this project off the ground and to getting this done in a real collaborative way with ATAP. So thank you for your time, and I'm happy to answer any questions. Go.
Hi, thanks for the presentation. Oh, this is loud. I had a question about, I think it was the IP2, the 7 megaelectron volt. The experiment where you irradiated an ear of a mice, and then you showed some graphs where you mentioned that there was a curious finding where I think the other ear was also showing some kind of results. I was wondering if you could kind of explain that part again. I was a little
Yeah, so
confused.
we irradiated the left ears of these mice, and they definitely showed this increased in thickening, sort of at the peaks at 20 days, and then it sort of resolves. Um, uh, we didn't see any of the thickening on the other ear. The right ear was completely normal, just by visual inspection or by doing the thickness measurements. But when we did our measurements with a very sensitive FTIR, so an infrared spectroscopy method, we were able to still distinguish the unirradiated ear of that mouse from a mouse that never seen any radiation. And so there could very well be a systemic effect. So you're radiating not just the skin, you're also radiating, for example, the blood that goes through that skin at the time of radiation. You're having not just a tissue response of that area, but you're having sort of a body response as well. Your body responds through being exposed, even though it was that ear. And that response might cause some changes. I would not be surprised in immune system infiltration, for example. And so there's probably an effect on the contralateral ear that is more systemic and not due to the, it's called an off-target effect, essentially, is what it's known in that field.
Yeah, thank you for the talk. So my question is related to the mice and then the studies at the ALS. When you delivered like a 50 gray dose to the mice, I believe they had a even larger response, and some of them had this huge response to the like the ear thickening.
Mhm.
I wasn't sure if that was a faster dose or a just a larger dose overall.
You mean on the two mice, specifically on the two mice that showed the larger response?
Any of them that had the
Any of
50
them.
gray.
So those were irradiated at Bella. So the instantaneous dose rate is the same for all the mice irradiated at Bella. And the
Okay.
median dose rate is, or the mean dose rate is also pretty much constant. So it's the same dose rate. It's just we applied a 50 gray dose, a higher dose total. So essentially more shots on the
Right.
mouse, on the
Okay.
ear.
And then I noticed you didn't some of the later analyses you didn't carry forward the 50 gray dose. Was that did you kind of determine 50 gray was too high?
It seemed a little high. We had, it's still preliminary data, so we haven't, if we wanted to do the 50 gray with X-rays, we could do it, but then do we also do the RBE correction on that? So then we would have to go 55 gray and then sort of the 40 gray, around 40 gray seemed to be the critical window where people see these skin effects in mice after radiation. So that's sort of why we focused on that.
Okay. Yeah. Thank you.
Mhm.
Really cool talk. Do you have any sense of where the protons are interacting within the cells? And my sort of thought is, which part of the cell is being damaged and how that impacts survival fraction and how that would change as a function of the energy of the protons or,
Great. Yeah, it's a good question. It's interacting. The particles basically traverse through the cell and are basically leaving a path of damage going through a cell. So it's interacting with everything in its path, really. The critical components, of course, are the nucleus where DNA is, and looking at DNA strand breaks, especially double strand breaks, being the most lethal form of DNA damage. So that's definitely one area. But protein damage is also, and lipid damage of the cell membrane can also play an important role in that response.
So can you disentangle what's happening based off if you look at this, like the clonogenic studies later on would tell you, oh, some DNA's damaged, they're not replicating, as opposed to if you like looked at the cell right away, you could tell, oh, these cells died because it just ripped through the walls or something like that.
Yeah, you can't really do that distinction, but some of the work we did with Corey using the peptides and the peptide modifications is just a single peptide. Could we do that at a much greater scheme and look at all the proteins in a cell, for example, we're not there yet. I'm not saying that we could do that right now, but could you look at protein modification and see, is there less protein modification in a cell after flash radiation compared to conventional radiation? Or we can also look at more immediately after radiation at DNA damage endpoints. So we could actually measure the amount of DNA damage that's being delivered on, inflicted on these cells after radiation exposure. And you could measure that flash versus no flash, and then correlate that to their ultimate survival at, like, when you grow them out for 3 weeks. Because obviously the survivors are the ones that you're measuring. You're not measuring any of the dead cells. They're already gone at that point.
Okay,
So,
thank you.
Yeah, those are great questions.
Okay, we have someone else in the room, and then I'm looking for anyone online. If you want to ask questions, you can raise your hand.
Hi. Just two quick questions. So the first one, how the flash effect, do you see it across a large number of different cell lines? It seems like the effect, to me, the difference between the tumors and the healthy tissue seems like it's a biological effect. It's something about the tumor. So is that seen across a wide array of cell lines, or is it specifically HeLa? And then secondly, the sort of oxidative damage studies you showed with the peptides, correct me if I'm wrong, but it seemed like when you had a hypoxic environment, there was less oxidative damage, which makes sense intuitively, but that sort of pulls in the opposite direction of what you're seeing in tumor cells, because those tend to be hypoxic. So curious, maybe ask you to speculate a little bit on where you think the effect is actually coming from.
Yeah, the oxygen effect in the tumor. Maybe Jamie, do you want to answer the question about the oxygen effect? It's a complicated answer. So the tumors, you're right, are already hypoxic. So that's why having less oxygen wouldn't actually, the effect of oxygen is negligible there, because they're already hypoxic. But in a normal cell, which is in an environment that is much more oxygen rich, that drop in oxygen that you might get when you flash irradiate a normal tissue, or basically soak up the oxygen in the immediate vicinity, and the diffusion rate may not be, there may just not be enough oxygen diffusing back into that area to cause more damage. Whereas when you're irradiating at very low dose rates, conventional dose rates, there might be this continuous coming in and out of oxygen and causing damage continuously, which you may not have when you do this with flash. It's so short that it's only the oxygen that's there at the time that can inflict the damage. And then you're already done with the exposure. So that might explain that. And then your other question was about the if you see this in a wide variety of cells. Actually, the flash effect is not seen traditionally in cells very much in the literature. It's mostly seen in vivo. And the reason we saw it might be because we had to develop these little cell holders, these metal little cell holders, and we had to clamp these like the day before we went up to Bella to get them irradiated. So those might actually not be in ambient oxygen. Those might be in a much more normoxic oxygen environment, so that the oxygen that you would have in tissues. And that's maybe why we're seeing this. And it's something that we've been wanting to explore by like drilling a little hole and putting an oxygen probe straight into those holders and seeing how the oxygen actually drops after you irradiate those things. So.
Anyone else? I don't see anyone online. So we want to thank Antoine
Sure.
Snijders
Thank you.
very much for talking.
Appreciate it. Thank you.